Okay, so thanks, Max, for the introduction, and thanks for everyone that tuned in for this tutorial. I will talk about modeling concentration-driven processes with PLUMED. And so to start, I will actually I would like just to jump in the session and recap a bit what I will discuss during this seminar. So in short, I will start by introducing why modeling concentration-driven processes with MD simulations and at the atomistic scale is challenging and why PLUMED helps a lot. And then I will recap what are the exercises associated with this session. After that, I will show and discuss a little bit of background and provide a few examples that are perhaps a little bit more interesting from the application point of view, but will be connected to the exercises in order to see where the application of some of the ideas that we'll start discussing today actually can lead. And then I will give a preview of the material that's provided with the tutorial and give a few hints about the resources available for this session. So to start, actually, I would like to define what a concentration-driven process is. And in this sense, especially in the context of molecular simulations and the type of simulations that we perform within this field. So by concentration-driven process, I mean a process that is initiated out of equilibrium in a metastable state, and where the driving force that leads to the transition between this metastable state to a more stable state, it's actually defined by the concentration in the metastable state. So to give an idea of this type of processes, I have a prototypical example of this type of phenomena, which is the nucleation of a droplet of a liquid from a supersaturated vapor. So in our case, the reactant is the vapor here, and it's metastable because its density it's higher than the equilibrium density at the pressure at the temperature of interest, and so this system evolves and forms, given enough time, forms a droplet of liquid. Now, in macroscopic terms, when we think about this problem, we have a well-established language to discuss this type of transitions, and this happens by essentially evolving along some reaction coordinate. Overcoming a free energy barrier, and essentially overcoming the free energy barrier leads to the formation of the stable state, in this case a droplet. Now, for a process like nucleation, the reaction coordinate is typically associated with the size of the droplet in some form, and the barrier corresponds to an important quantity in the field or for this type of processes, which is the critical nucleus size. Now, when we try to highlight and study this type of processes in a molecular simulation, which is constrained, so it contains a fixed number of particles, actually what happens is that we observe an evolution over a free energy surface, which is distorted with respect to the macroscopic counterpart. So typically, what happens in a molecular simulation is that as the droplet forms, so as the system moves from no droplet on this end of this reaction coordinate to a droplet on this other end of this domain, essentially the concentration in the metastable phase changes. And this change in concentration leads to a difference, a qualitative and quantitative difference in the free energy associated to the transition. Now, in this context, we know how to deal with the process, and we can assess what are the effects on the process itself. However, one needs to be able to recognize that this is happening and be able to investigate the effects of this type of confinement. So in this context, actually PLUMED comes in and helps in a few different ways. The first and the most typical one is that PLUMED, is an engine that allows to perform enhanced sampling calculations and free energy calculations. So a typical application for PLUMED in this context is for the calculation and the sampling of these free energy barriers. So essentially, by enhancing the transition from the metastable state to the formation of the stable state, exactly like in any other enhanced sampling application. A second context, which has also overlaps perfectly with typical applications of enhanced sampling in other fields, is in the definition of the reaction coordinate here. So PLUMED comes in and helps in the definition of a reaction coordinate that is able to actually connect a metastable state like this to a stable state where a droplet is formed in this type of problem. However, there is a third area which will be the focus, especially of exercise 2 and 3. In a second, we'll discuss them, which is essentially using PLUMED to introduce forces that allow us to bridge the gap between a finite size problem, so a problem that evolves a constant number of particles and therefore evolves on this modified, on a distorted free energy surface, with a problem that with its counterpart in a macroscopic system, so essentially evolving without that type of constraint. So the exercises and examples that we'll give you in this session are related essentially to these two parts. So the use of PLUMED to defining reaction coordinate for this type of systems, and the use of PLUMED to bridge the gap between macroscopic and finite size simulations, in particular in order to maintain or to study the evolution of this type of processes under a constant driving force. And before I start, I would like to acknowledge the people that is active, especially in this third branch of this endeavor of using PLUMED in this context. In particular, Claudio Perego, who developed the method that I will discuss, and Tarak Karmakar, who's been using it more recently in very interesting applications for nucleation, free energy calculations, and so on. Ozgur Yazaidin, who is a colleague at UCL and has been using this type of ideas to investigate fluxes through porous media. Aaron Finney, who is a postdoc here at UCL and is using this to investigate electrodes and absorption on electrode materials. And then of course, Gareth, because Gareth is the main player behind the implementation of some of the ideas that allow us to compute effectively reaction coordinates in this context. So in order to start, I would like to summarize a little bit the 4 exercises that we would like to propose, and then I will jump into some examples that perhaps we'll be able to connect with these exercises. So the first exercise will look at a trajectory in which we observe a nucleation event, and we will the objective of the first exercise would be to compute a reaction coordinate that is able to describe the evolution of the system. So in particular, that is able to count the number of atoms, in this case, that will transfer from the metastable vapor phase to a liquid phase. The second exercise will be based on the analysis of the same trajectory, but essentially we'll look a little bit deeper into what happens in the trajectory, and will actually be based on analyzing clusters that form, how many of them are there, how big they are, and how they contribute to the evolution of the system. And in this case, the key topic will be the application of depth first search clustering, which is a method that, again, that Gareth especially contributed implementing in PLUMED. And then the third and fourth exercises will be focused on the use of a method called SimuMD, which is the method that originally Claudio Perego developed, based on essentially introducing forces that allow us to model this evolution at constant concentration. And I will show 2 different flavors. The first will be simply based on essentially investigating a diffusive process. A diffusive process that takes place within a simple system. The second instead will actually look at using SimuMD to study the growth of a liquid phase under constant concentration. And in the last one, the exercise 4, one should actually be able to, or you will actually learn how to combine ideas from the previous examples in order to analyze and carry out this type of analysis. So since it's an introductory lecture, just to give an idea of why these ideas are important, I wanted to provide a few examples. So the first example I want to give is about crystal nucleation. This comes from a paper that Aaron Finney recently published. And it's an example in which essentially we are using ideas that are very similar to those that will be developed in example 1 to count molecules that, in this case, not molecules, ions that aggregate and form clusters and little crystals in a simulation from solution. And in this case, we are projecting the dynamics as a function of 2 descriptors that have been computed with PLUMED using this type of ideas. One descriptor is called N and correlates with the local density of these clusters. So essentially, the higher N, the higher the size of the cluster, regardless of its order. And the second descriptor is called N of Q6, so correlates with the local order of these clusters. And today we will see something that is very similar to N, but essentially the ideas that we will develop can be applied also to this type of descriptors that include some notion of order inside of their formulation. And in this case, it's important for us to be able to compute this type of descriptors, because the evolution of a crystal that emerges from solution, even for a system as simple as sodium chloride, is far from being simple. Actually, it shows a complex dynamics and a complex evolution, and depending on the driving force, the evolution of the nuclei that emerge in these conditions may involve, for example, multiple steps. And in this case, we find that in conditions that are actually supersaturated, but not beyond the spin-order decomposition, nuclei like this one actually emerge by first forming a dense core and then ordering within that core. And again, it is instrumental for us to be able to describe this process quantitatively, to compute variables that are very similar to the variable that we will discuss in exercise 1. The second big idea within this tutorial will be about essentially mimicking an open system. And here I would like to again go back to the second type of problem that will be essentially at the heart of examples 3 and 4. And in this case, basically, I would like to discuss what happens when we have a system like a simulation box, which is sketched here, where we have a region of the simulation box that represents a sink, like a crystal, and a solution that provides material to this sink. Now, in a situation like this, in a macroscopic system, we would see that the growth of this crystal, so that this front moves, so it grows under the effect of a concentration that never changes, because typically a crystal is in contact with a reservoir that is much, much larger than the volume that we are able to model directly in a simulation. When we perform simulations of crystal growth instead at the atomistic scale, what we observe is that the concentration in the liquid phase changes as the system moves along and progresses. And so here is where this idea of SimuMD, or constant chemical potential molecular dynamics, comes in and helps us essentially mimicking an open boundary system. So in this case, this will be the open macroscopic counterpart of a SimuMD simulation, where we have a crystalline phase and interface. And then in solution here, we have a transition region where the concentration in the liquid phase relaxes, and then it reaches a bulk. In a perfect simulation that is able to mimic open conditions, this bulk would be at constant concentration for an indefinite distance from the interface. In reality, we know that this is not the case. So what SimuMD does is essentially it couples this type of system with a reservoir, which is a physical reservoir. It's present in the system that we model. But essentially, it couples the system by introducing some forces that act at the boundary between the control region, where we want to maintain the concentration at a certain value, mimicking a bulk condition, with the reservoir where we have an arbitrarily large number of particles that we can draw in to control this process. Now, this happens due to the fact that we are able to apply forces that are indicated here as F mu. And these forces depend on the distance of the concentration, of the instantaneous concentration in the control region with a set point distance. And this expression for the force is applied through a switching function that acts in this region of the simulation box with the ions that essentially are in this region. So this is the formulation of the switching function. And the point I want to make here is that this is made possible, and it's made possible by using PLUMED, because essentially we can express the density of any species as a function of the coordinates of the atoms in the system. And in particular, here we use a function of the position in Z, where Z is the coordinate that goes away from the interface. And this function is a switching function that tends to 1 if the position of a certain atom is within this region, and it's 0 otherwise. So essentially here we are exploiting the ability of PLUMED, or facilitating the application of forces as a function of the Cartesian coordinates that characterize the system, to apply a force that has the physical meaning essentially of maintaining constant the composition in a region that models the bulk for our system. So this is important in order to give a little bit of background. This is important because the growth of this crystal phase, or the rate at which atoms are removed from this system due to the fact that we have a process of crystal growth in this case, depends on the composition in this region. And this is so because the chemical potential, the difference between the crystal at a certain temperature and pressure and a solution, crucially depends on the concentration in the solution. So if you write the chemical potential of the crystal without entering into the details, the point is that the chemical potential of the crystal will depend on the solubility of the material that forms this crystal at a certain temperature and pressure, and so it's independent on the composition in the system's bulk. However, the chemical potential of the liquid bulk will depend crucially on the activity of the species that is being removed from the simulation. The activity is a function of the composition. Once again, one can write it as an activity coefficient that multiplies the molar fraction, which are all quantities that essentially depend on the local composition. And when one writes a difference in chemical potential between the liquid and the crystal, essentially one can see that this difference is controlled by the concentration in the liquid phase. So being able to control the liquid phase concentration has the very practical effect of essentially allowing us to control the difference in chemical potential between the liquid phase that is driving the process evolution and the crystalline phase. So to show an example for this, I have this example that comes from the paper from Claudio that introduced this method. So in this case, we have a urea crystal slab. So this is the sink of urea molecules. And this urea crystal slab acts as a sink because in these conditions of concentration, its chemical potential is lower than the chemical potential of the solution. So molecules in solution want to actually absorb and become part of the crystalline phase. So if we perform a simulation in the NPT ensemble without any application of SimuMD or any application of special sampling ideas, basically what we observe is that the number of molecules in the crystalline state increases until it reaches a plateau. And this evolution happens against the background concentration in the region of the simulation that represents the bulk that is never constant. So the size of the crystalline phase increases, the composition or the molar fraction of the solute that is removed from solution decreases, but the evolution never really takes place at constant composition. Instead, if we apply SimuMD, applying forces in this region and essentially controlling the concentration in these control region areas of the simulation box, we are able to govern the rate at which atoms are removed. And we are able to observe the evolution of the system in out of equilibrium conditions because the difference in chemical potential between the crystal and the liquid is constant, but at steady state. So, for example, if we apply SimuMD with a target concentration in this region, so with a molar fraction around 0.05 in this plot, we see that essentially after a short equilibration, the system evolves at constant composition in the bulk, and atoms are removed at constant rate. Well, this method has a problem because the reservoir associated with it is physical. So at some point we run out of molecules in the reservoir, and what happens is that the method cannot cope, and we get back to a flat plateau. In other words, we cannot escape the mass balance for a simulation box that looks like this, even if we have forces acting between the reservoir and the physical part of the system. However, we are able to sample the system reliably in out of equilibrium conditions, but at steady state. And this is very important to understand the physics of the growth process. So for example, in this case, if we change the concentration, we raise it, we observe that the slope of this curve changes, and again, and for example, one can study systematically what happens to the growth rate, which is this slope, the slope of this curve as a function of composition, and discover that for a diffusive growth process like this one, they're linearly dependent, which is an expected result, but it's also a nice confirmation that we can indeed perform this type of study. Again, the success of this type of method for crystallization problems is heavily dependent on the physics of the crystallization process itself as well. For example, we investigated also a second urea crystal surface, which does not grow through a diffusive process, but it's characterized by activated events. But in this case, the activated events have a characteristic time which is sufficiently short that we can anyway sample them directly with unbiased dynamics using SimuMD. And we observed that also in this case, we obtain a behavior that is consistent with a steady state out of equilibrium evolution. So in this case, I'm comparing the evolution of the number of crystalline molecules in the case of SimuMD, the top panel here, and in the case of a normal NPT simulation here. And here the figure of merit is the fact that in this case, these jumps are distributed in a way that is completely independent of time. So essentially, we can predict the distribution of these activated events as if it didn't depend on time. While in the case of an NPT simulation, since the driving force changes as the simulation goes along, also the probability of observing the next nucleation event that leads to growth decreases. So you see that qualitatively here, the steps get longer and longer. Instead, in this, if we get enough statistics out of these simulations, we see that the distribution is consistent with a single characteristic frequency, which is what we did here more quantitatively. And okay, this also allows us to qualitatively assess what is the free energy landscape associated to the process and what is the characteristic barrier for nucleation events on this type of surfaces. So the last exercise, the final exercise, will deal with this type of processes where we have a concentration-driven removal of molecules or particles from a simulation box. But there's also another potential application for this type of simulations. So another paradigmatic example of a concentration-driven process is a diffusive process. So a process of diffusion that is driven by a difference in concentration between two different regions of a simulation box. And in this case, I'm showing results that come from a paper from Aydin, who's a student at UCL, supervised by Ozgur Aydin. And in this work, basically the idea, the main idea is that we're able to model a diffusive flux across a MOF membrane by imposing a difference in concentration between the two sides of the membrane without applying any external force to the membrane itself. So here, Claudio's idea of SimuMD actually applies in an asymmetric fashion. So we have, again, a membrane, a transition region where the concentration relaxes to the bulk volume, the controlled regions where we are modeling a simulated bulk mimicking open conditions. But in this case, we have an inlet and an outlet control region. So we have two control regions which have two different target concentrations. And then we are applying the forces as usual, and we have a reservoir. So in this case, we have no artificial forces in this internal region of the system. And within this region, the diffusion process is completely dominated by and is completely dictated by the difference in concentration that is observed across the membrane. So as you can see, the added bonus of this type of variant of this method is that in this case, essentially, the removal of molecule is not limited by the size or the extent of the reservoir, because essentially molecules that absorb on one side of the membrane are desorbed and accumulate in the reservoir on the opposite side. And since we are operating under periodic boundary conditions, essentially we generate a steady state flux across the periodic boundary the periodic boundary that is orthogonal to the membrane surface. Okay, so the third exercise in this session will actually be about essentially performing this type of simulation for a very simple fluid in the absence of a membrane, because essentially every exercise is targeted to essentially be able to be reproduced on a small machine, so without significant computational cost. So we try to apply this idea without the extra burden of having to model an interesting material in between the two regions. Okay, there is one final application that I want to raise. These are the results. Okay, there's one final application that I want to raise about the use of SimuMD in the context of this type of processes, and this application comes from my group and from the work of Dr. Aaron Finney. And in this case, basically, we're using SimuMD to study the absorption of a multi-component liquid phase onto surfaces, and in particular onto graphite surfaces. So here we have a solution where we have sodium and chloride in water in contact with a graphite interface. And in this context, even though it might look like the situation is different than the sink source problem that we've discussed in the previous cases, it's actually subtly similar in the sense that different ions have a different tendency to accumulate at the interface between solution and the solid. So the affinity of the different ions here represented in blue and cyan, sodium and chloride, is slightly different. And this means that if the simulation box is small, which is typically always the case for us, the preferential adsorption of one of the species will lead to essentially a charge imbalance in what we model as the simulated bulk. So in other words, in the region that we model as bulk, we will find that the system is not electroneutral, as it should be instead if it was in contact with a macroscopic reservoir. So in this context, we are using SimuMD to control the concentration in these regions and to making sure that essentially electroneutrality is always maintained regardless of the concentration at which these simulations are performed. And this type of result is shown here in the corner, where we monitor the concentration in the bulk of sodium and chloride for every for simulations performed across a wide range of concentrations. Now, being able to perform this type of analysis is important because it allows to discover interesting features of the physics of these problems that would not be as clear-cut if we were not able to maintain an electroneutral background for this type of simulations. So in this case, Aaron discovered that the structure of the double layer, so the structure of the region close to the interface where these ions accumulate, is far from being close to ideality. So an ideal classical picture for this type of system would actually see a size of the double layer decaying exponentially with the concentration. Instead, we observe that due to different processes, such as, for example, the formation of local structures in the concentration profiles, we we a non-monotonic behavior of the double layer size, of the double layer thickness for this type of processes. And this type of observations have also been backed up by experiments. So I'm showing here a number of electrochemical properties of the double layer that were computed in this paper by Aaron. But the reason why I'm showing this is not really to go into any of the details, but just to show that this ability of maintaining an electroneutral background for this type of systems is key actually to learn and quantitatively compare these simulations with experiments. Okay, the final example slide that I have that connects to the exercises is still coming from this paper, and it's coming from the fact that once we looked at the results from these double layers a little bit more in detail, we discovered that at least within the remits of the model and the simplifications that we are introducing in the system, there are structures that form in the double layer. So we can observe that, for example, we can compute the average coordination number between anions and cations as a function of distance, which is something again that relates quite significantly with the quantity that we will derive in the first exercise. And we see that essentially we have a peak close to the interface, which is well defined. And looking at the diffusivity of these ionic species in these regions, we see that there is a slowdown, and this slowdown is dependent on concentration and on distance. But more interestingly, we see that if we are able to characterize the population of clusters within the double layer and within the bulk that is in contact with the double layer, we see significant differences between the two. So here I'm showing a plot that in principle you could produce also after today's tutorial, where on the x-axis we have the number of clusters that are observed in a system, and on the y-axis there is the number of ions in the largest cluster. And there are two data sets on this plot. One data set is represented by squares, which are here on the bottom, and the second data set is represented by circles. And the color code goes from low concentration to high concentration. So as we move from blue to red, we go from low concentration to high concentration. So what's interesting about this set of data is that as we move from low concentration to high concentration, of course, the number of clusters increases, but we have a significant departure of what happens in the double layer in terms of the size of the largest cluster. So basically, in the double layer, we have that the size of the largest cluster observed departs from what you would observe in the bulk of a liquid phase. And this is interesting because it's suggesting that the interface is actually catalyzing the formation of large connected structures that may be precursors for a crystal phase. Okay, so after this round of examples, is there any question? I saw a.
Can I ask one, Matteo?
Yeah. Yes.
I was curious on this last application, if you are using periodic boundary conditions and how critical is that for long-range electrostatics interactions?
So that's a good question. We are using periodic boundary conditions, and what we are observing so far is that the reservoir, which contains ion, actually screens interactions between the images. But I agree that that is a critical point, especially if you want to move towards application where you introduce a charge on the surface. And so, for example, in that paper, we also analyzed for one specific concentration what happens at different surface charge levels. But to perform something that would be more quantitatively comparable with experiment, for example, one would like to have a constant potential rather than a constant charge. So it's a critical point. We don't think we don't think it plays a big role for our system due to the screening of the reservoir, but it's indeed an open area.
Okay, thank you. There's a question from the chat also. I can see.
Yeah.
You can see it as well.
Yeah, Simone is. Yeah, he's asking if the PBCs are in the XYZ. Yes, they're in the XYZ, but the forces are acting only on Z, on the Z coordinate. So they only. So this is also connects to recent developments led by Tarak, where basically all the applications that I've discussed now and that are in the examples that I'll show in a moment essentially imply planar symmetry for the system. So the sink or source as a planar system, symmetry in the system. You could make different choices. So there is a spherical variant for this method that has been used to model nucleation, where you have a sink for the atoms that become part of the new phase that is surrounded by a spherical region that acts exactly like the control regions in our setup. But in both cases, the periodic boundary conditions are in X, Y, and Z. Okay. So what I wanted to do now was to go back and look at the exercises a little bit more with having given this background information. And so in order to do so, I will do two things. The first, I will refer to the web page where the tutorial is hosted, and then I will we will give a bit of a sneak peek into the data available for that exercise. So the first exercise, as I mentioned, focuses on essentially defining a variable that characterizes the transition from a metastable state to the formation of a stable droplet in a supersaturated vapor. And on the tutorial, this first exercise is described here, where basically the objective is to learn how to use a multi-colvar. So a collective variable that is based on the properties of a distribution of collective variables computed on a population of atoms in this case. So in brief, what this first exercise describes and asks to do is to essentially define a liquid-like criterion for the molecules that are present in the simulation box. And then by computing that liquid-like criterion, essentially asks to follow the dynamics of what happens in the simulation box. And there is a little bit of a suggestion also on how to run a PLUMED driver to analyze the trajectory that is available in the data set that you can download from GitHub at this address. And just to have an idea of what I'm talking about, I have this is the system that you will be playing with exercise number 1. And this is the final state. The initial state is a supersaturated vapor. And essentially what happens in this simulation is that as the density is larger than the equilibrium density for the system at this temperature, the process evolves and clusters form and aggregate into a single spherical droplet. So if we go back, at this point in time, there are no, or there's a very little number of liquid-like atoms based on a criterion on the number of nearest neighbors that every atom has. But as the simulation evolves, you see that clusters form, and then these clusters merge into a large spherical droplet. So the first exercise, go back to the web page. The first exercise will be based on defining a multi-colvar based reaction coordinate for this system that essentially classifies an atom as liquid when its number of neighbors exceeds a threshold, which in this case is 5. For every one of the exercises, there is some suggested activity that you can do essentially based on what has been made available with this, or following the suggestions of this first example. So here, for example, what I'm suggesting, whether using similar ideas, you can classify all the atoms that are on the surface of these clusters, and whether one could directly estimate in PLUMED how the surface to volume ratio of these droplets go as the system goes along. Now, the second exercise is inspired by the work that Aaron did on these clusters close to graphite interfaces, but is still based on the trajectory that you've seen. And the objective of this second exercise is to learn how to use DFS clustering within PLUMED to compute the number of clusters that are formed, the size of the clusters, and the size of the clusters, essentially. So what DFS is and does is essentially a method which based on graph theory to compute the number of clusters and the number of elements in each cluster by defining a contact matrix, essentially. So in this case, we compute again a coordination. We use again the coordination number action that was defined in the previous example to identify the liquid-like atoms. And then we identify which one of these atoms are close enough so that we could define an adjacency matrix for the system that identifies, essentially that defines a graph of connections between all the atoms in the system. And then what DFS does is identifies the connected components in that graph. And the largest connected component is the largest cluster in the system. So essentially, with these actions, we are computing the largest, the second largest, the third largest, the fourth largest cluster. And then essentially, we are printing out the information that we have with this print statement. Another aspect that is interesting to follow the dynamics of this process is the number of clusters in this type of process. And this is computed using this cluster distribution statement. Okay, so in this case, essentially, what this second exercise will do, essentially, will give you some tools to interpret the trajectory in order to quantitatively analyze what happens within the system. The third and fourth examples, instead, they depart a little bit from these two, because these two exercises were essentially based on using PLUMED as a standalone code using driver, essentially. But for exercise 3 and 4, instead, the suggestion is to actually run a short molecular dynamics simulation using GROMACS patched with PLUMED and using SimuMD. So in this case, we have two examples, and the first one in exercise 3 is the simplest one, that is simply based on imposing a steady-state diffusive flux using SimuMD. So again, in the material that's available, there are some links to the GitHub page where we are hosting the source code for SimuMD, where there are some details on the keywords that are used. But I want just to give an overview on this case. So here we define a group of atoms. So in this case, it's all the atoms in the system. And then we define two regions, two control regions, one left and one on the right. So in this case, again, we have a planar symmetry with Z being the position coordinate that we're interested in. And the left action here defines essentially a region on the left side of the simulation box, while the right action defines a region on the right end of the simulation box. And what these two actions do is essentially to compute, to count the number of particles that end up in these regions. And then by defining these two restraints, one on the left and one on the right at different values, we are able to essentially restrain the concentration within these two regions to two different values, one high and one low. And again, this is just some logging of the data. So I want to give you an idea of how a simulation like that could look like. And so this is a simulation box for this type of system. Again, the number of atoms is very small. So it's only 1,000 atoms. This would be consistent with the left and right. Okay. And in the initial conditions, every atom in the system is in the reservoir. The reservoir goes across the periodic boundary conditions in the Z direction. And essentially, as the system evolves, after some equilibration, you see that it will develop a steady concentration field, essentially a steady state concentration field, where essentially we are governing. So the right side of the right region is on this side, while the left region is on this side. The concentration in this region goes to a steady state high value. The concentration in this region goes to a steady state low value. There are no spurious forces in this region, but we have the establishment of a diffusive process and a stationary concentration field. I want just to show quite. Let me see if I can. Okay, this is the concentration, the number density as printed by the code. This is on the left region, which is at high concentration, and this is the one on the right region at low concentration. And you see that there is an equilibration period, but after equilibration, essentially the concentration in the two regions remains constant, and the composition field becomes stationary. Okay, so this is for exercise 3. And as additional activity that I was suggesting for exercise 3 was to perform simulations at varying concentration differences and computing the concentration gradient across the simulation box as a function of the concentration difference. These are, for example, especially in the second part, many ideas related to the use of multi-colvars can actually become very, very useful. And the final exercise is essentially exploiting a similar idea of using SimuMD to maintain the composition of a fluid phase constant, but in this case, to actually promote the absorption of or the condensation of fluid on an amorphous surface. And in spirit, this is very similar to a crystallization or to an adsorption process. And again, in this case, this is how the system looks at the beginning of, so in its initial conditions. This yellow region is maintained fixed. So essentially to minimize the computational cost of these simulations, this is simply acting as a completely inert support. But the blue particles here that at the beginning of the simulation are all in the reservoir are subject to the SimuMD type of algorithm. And what happens here is that again, once the simulation starts, we establish again a composition field that is steady. And since the composition on both ends is larger than, essentially the composition on both sides of these surfaces is such that the chemical potential of the vapor phase is higher than the chemical potential of the same species absorbed on this surface. What happens is that the adsorption process proceeds, and these interfaces essentially act as a sink and remove molecules from the surface, from the simulation boxes, and they do so at a constant rate. Also, in this simulation, I wanted to feature the limit of this method, which is when your reservoir empties and the number of molecules that you have set up at the very beginning is not sufficient to cope with the longer process. That said, the evolution between the onset of a steady concentration gradient and the fact that the reservoir becomes empty, it's a steady state conditions. Also, for this case, I think I had already the that's again, this is what happens to the density in the two controlled regions. So there is a very short equilibration period, and then the two regions evolve. So the system evolves under steady state conditions. So in this region where everything seems constant, in reality, what happens is that adsorption is taking place, but it's taking place against a background that is a constant concentration. And then when sufficiently large number of atoms has been removed from the reservoir through this steady state process, what happens is that the system is not able to cope anymore with the evolution of the process. But the key aspect of this is that in this region, we were able to sample an out of equilibrium process under steady state conditions. And so again, the information that's given in this exercise 4 is necessary it's available to replicate this type of simulation, or to analyze the existing trajectory that is available again at the same GitHub repository. But the interesting thing is that if one wants to play around with this idea, many of the ideas of the previous exercises can also be used. So, for example, you could use the ideas that were developed in this first exercise to actually count the number of molecules that transition to a condensed phase during the simulation, and trying to prove that that process is actually happening at the steady state conditions. And also one could play around with the conditions in the controlled regions and the densities that drive this adsorption process to study how the adsorption depends on the concentration, how the adsorption dynamics depends on the concentration in the background homogeneous phase. Okay, so these are the four exercises. And all the files for these 4 exercises are available at this GitHub. The description of the tutorial is available on the Plume website. Some additional resources that might be useful if one is interested in investigating these ideas to sample these type of problems can be found again on the SimuMD GitHub, where there's also an extensive discussion of the options and keywords associated with SimuMD action. And then there are some additional examples where both SimuMD and also clustering ideas were used in a much more complex setting with ordered domains and more complicated corrective variables. And those are available through PLUMED. And again, we have support through Slack, as you know, and the correction will be next week on Wednesday, I think at the same time, right, Giovanni?
Yes. So, any questions, comments? Okay, good. So